So my name is Julien Spatazza, and I'm the head of pharmacology at BrainEver. So first of all, I'd like to thank the organizers for giving us the opportunity to share BrainEver's recent progress on developing HengRen1 homeoprotein as a novel therapy for ALS. Oops. So BrainEver is a Paris-based biotech company that's building a homeoprotein-based therapeutic platform to tackle neurodegenerative diseases. So as of today, we're actively working towards bringing human HengRen1 protein therapy into the clinic for the treatment of ALS. In the near future, we're hoping to build on internal data supporting the use of OTX2 homeoprotein for the treatment of various eye diseases. And eventually, we're hoping to move on to other indications as well. So homeoprotein transcription factors are key transcriptional regulators with well-established developmental functions, including the control of morphogenesis, lineage decisions, and cell differentiation. But in addition to these well-documented cell-autonomous activities, homeoprotein transcription factors also have the really peculiar capacity to be transferred between cells, which is represented here, allowing them to exert a wide range of functions in a non-cell autonomous manner. So this property was discovered by Alain Prochian, our founder, and his colleagues. And what they also discovered, they also managed to map within the DNA binding domain of the homeoprotein transcription factors, the sequences that are responsible for both secretion and internalization. So the DNA binding domain is highly conserved within this huge class of transcription factors, thus indicating that homeoproteins constitute a novel class of signaling molecules. So given that the expression of homeoproteins is maintained in the adult central nervous system, the work performed by our group has really focused on trying to understand the functional significance of this maintained expression in the adult, and notably in the context of neurodegenerative diseases. So an example of homeoprotein transfer was recently observed in the adult spinal cord, where HengRen1 is expressed by V1 interneurons, here in red, and a subpopulation of Renshaw cells, here in blue, that are both located in the vicinity of chat-expressing motor neurons in the ventral horn of the spinal cord. By combining RNA scope to reveal the presence of HengRen1 mRNA, here in red, to an immunolabeling for HengRen1 to reveal the protein, here in green, we could observe a neuronal population that contains the protein without expressing the mRNA. We went on to confirm that this neuronal population corresponds to chat-positive motor neurons that are located right next to V1 interneurons that express HengRen1, thus suggesting the possibility that HengRen1 is transferred from the V1s into nearby large motor neurons. To assess the role of HengRen1 in adult spinal cord physiology, we took advantage of a mouse line that's lacking one HengRen1 allele. These animals were tested for various behavioral and motor tests that include the grip strength test, the inverted grid test, and the extensor score. And this was done at various postnatal stages, as indicated here. For simplicity reason, only the extensor score results will be presented today, but it's worth mentioning that the various tests that we use, they all converged in indicating that these animals display muscle strength deficits, and we'll cover that in just a second. We also analyzed these animals at the histological level. We looked at the NMJ, and we also used spinal cord tissue sections to notably evaluate motor neuron density in these animals. So by comparing wild-type animals here in gray to NGRN1 mutant animals here in dark green, what we could see is a progressive muscle strength deficits in these animals, which is associated to a progressive denervation of the neuromuscular junction. And these two parameters precede the selective loss in alpha motor neurons, thus suggesting that alpha motor neurons in the NGRN1 mutant animals experience retrograde degeneration. In an attempt to evaluate the contribution of NGRN1 transfer in motor neuron survival, we developed viral tools to overexpress a secreted single-chain antibody directed against NGRN1 so that extracellular NGRN could be captured prior to its internalization by motor neurons. And this is schematically represented here. So the construct encoding for the single chain was packaged in an AAV8, which was injected in wild-type mice at the age of 1 month. And the injected animals were followed up to 6 months post-injection. But the first thing that we did was to confirm that this manipulation truly results in an impaired NGRN1 transfer into motor neurons. And sure enough, this is what we observed. It's illustrated here and quantified here, where the animals that overexpress the antibody have dramatically reduced NGRN1 levels in motor neurons compared to non-injected animals here in gray, or animals that overexpress a mutated form of the antibody. What is the functional consequence of this impaired NGRN1 transfer to motor neurons? Well, we observed, again, a progressive muscle strength deficit in these animals, which was associated to denervation of the NMJ, and again, a selective loss of alpha motor neurons. So taken together, these data demonstrate that NGRN1 is really key for motor neuron survival, which is reminiscent of previous work from our group showing the important role of NGRN1 in the survival of a different neuronal population, which are the dopaminergic neurons located in the substantia nigra. So since extracellular NGRN1 protein appears to exert a trophic role on motor neurons, we decided to evaluate whether the exogenous administration of human NGRN1 protein could be used to rescue the deficits displayed by the NGRN1 mutant mice. But first, we turned to wild-type animals, which we injected IT at lumbar level L5 with the protein in order to get a sense of its biodistribution in the spinal tissue. As illustrated here, 24 hours post-injection, human NGRN1 protein detected here in red was seen to be pretty much exclusively captured by chat-positive motor neurons in the ventral horn of the spinal cord. And this is quantified here, thus suggesting the existence of specific NGRN1 binding sites at the surface of motor neurons. We went on to perform a pharmacokinetic experiment to follow the fate of the protein within the spinal tissue post-injection. And the results here, what we see is a pretty rapid accumulation of the protein within motor neurons as early as 6 hours after the injection, and a pretty rapid clearance as well of the protein, as illustrated here at 3 days post-injection. NGRN1 levels in chat-positive motor neurons are pretty much back to baseline levels. So what happens if such an injection paradigm is applied to mutant animals? The first experiment that we performed, and that is not represented here, consisted in the injection of human NGRN1 in mutant animals at the age of 3 months, and the animals were evaluated 6 weeks later. And what we could see is a full rescue of the behavioral deficits displayed by these mice, and also a full rescue of alpha motor neuron survival. So these results were encouraging, and they led us to design this second experiment, which is highlighted here, and that I'm gonna present on this slide. So here we had two groups of animals, a red group where the mutant animals were injected only once and followed for up to 6 months, and the idea was to determine how long-lasting a single injection of human NGREL1 can be. And the second group was the blue group, where the animals were injected twice. So first time at 3 months of age and a second time at 6 months of age. So what are the results? So the red group, a single injection essentially brings back the mutant animals to wild-type performance levels for approximately 3 months prior to a progressive decline in muscle strength. So now what happens if a second injection is performed just prior to the decline? So this is the 2 injection group, the blue group. What you can appreciate is that the performance of these animals is maintained to wild-type levels for an additional 2 months. And this is corroborated by the histological analysis, where we notably see that the animals that receive 2 injections display at the very end of the experiment a totally normal amount of surviving motor neurons in the ventral horn, comparable to wild-type animals. So taken together, these results clearly indicate that NGREL1 has a neuroprotective effect, that it's fairly long-lasting, and that this neuroprotective effect can be prolonged via a second injection. So as a summary for this part, NGREL1 appears key for motor neuron survival. Mice that are lacking one NGREL1 allele, they display neurodegeneration, which can be prevented through the exogenous administration of human NGREL1 protein. Now, importantly, in wild-type mice, the sole neutralization of extracellular NGREL1, as illustrated here, is enough to induce an ALS-like syndrome. So we then wished to evaluate the activity of human NGREL1 in ALS models. And for that, we turned to patient-derived iPS cells to generate motor neurons. Specifically here, we've used an iPS line from a symptomatic patient harboring a C9 North expansion, and we also used its corrected isogenic counterpart. So the motor neurons were cultured within these microfluidic devices that are represented here. So the somas are here, and the axons of the cells are kind of forced to grow within those microgrooves through the application of a growth factor gradient in this red compartment. Once the axons reach the red compartment, they can be imaged on a microscope, and we can quantify the neuritic outgrowth of these cells. So the first thing that we did was to compare ALS and control cells, and what we observed is a reduced neuritic outgrowth for the ALS cells. Now, importantly, what we observe is that by simply applying in the axonal compartment NGREL1 onto ALS cells, we could rescue the neuritic outgrowth deficits that these cells normally display, as illustrated here and quantified here on this graph. So it is worth mentioning that these findings were extended to other iPS lines harboring various mutations of familial ALS. And in all these assays, NGREL1 was systematically found to be active and to be able to restore normal neuritic outgrowth, which suggests that NGREL1 could thus be active in various genetic forms of the disease. We went on further to evaluate whether human NGREL1 could be neuroprotective in vitro. And for that, we teamed up with Eva Hedlund, who's located in Sweden at Stockholm University. And we designed a simple oxidative stress-induced degeneration model where the, again, patient-derived motor neurons were cultured, pre-treated or not with NGREL 1 for 24 hours prior to a challenge with hydrogen peroxide for 2 hours. The readout was cell viability. The results are presented here. What you can appreciate is that in the absence of NGREL 1, all the cell tested here were dramatically impacted by the hydrogen peroxide treatment. But importantly, in the presence of NGREL 1, we could see a dramatic rescue in the viability of these cells, thus showing that ALS human motor neurons can be protected by human NGREL 1 treatment from oxidative stress-induced degeneration. So what is the presumptive mechanism of action for NGREL 1? So we know that in aging and also in neurodegenerative contexts, the sustained oxidative stress has an impact on the heterochromatin that makes possible the activation of line 1 retrotransposons that are normally repressed and that are found elevated in patients. This leads to DNA strand breaks and to cell death. So what I try to highlight on this slide is a few aspects of the activity of human NGREL 1, which we know of, and some of them have been documented and published, and that we think might be important here. So in motor neurons, we show that NGREL 1 regulate the expression of P62, which is a key ALS gene. In cultured motor neuron as well, we could show that it promotes the translation of STATMIN2 mRNA, and in a totally different context. We've shown that NGREL 1 represses line 1 activation, maintain the heterochromatin, and also promotes the expression of GAT45B, which is a DNA demethylase, which we think is really at the center of this long-lasting effect that we obtain in our system and also in other models, notably of Parkinson's disease. So we think NGREL 1 is really a multifaceted drug with the promise to be active on multiple forms of the disease. To wrap up, I just wanted to take this opportunity to share with you some preclinical data obtained in non-human primates and that support moving into clinical studies. So this is a distribution study that we performed following the IT injection at the lumbar level of radio-labeled human NGREL 1 protein. We use PET imaging, and this was done in collaboration with Misha Papisov at the MGH. We see pretty rapid distribution of the product in the spinal tissue and retention up to 6 days post-injection, which is the latest time we've evaluated so far. This relative distribution graph clearly shows that the retention is pretty much exclusive and specific to the spinal tissue, with very limited detection of the product at the periphery. We went ahead and performed a preliminary tox study in non-human primates as well, using IT lumbar injection. 2 doses were tested with a dose that's equivalent to 10x of our optimal active dose. Some animals received 3 administrations, and we had a time interval of 2 weeks between each administration, and the take-home message is that no safety signal were detected. This is the current status of our program, just to indicate that we're currently preparing for the manufacturing of our GMP toxic clinical batch, preparing our main GLP tox study, and also developing our biomarker discovery program, notably through a collaboration with Neurodex to leverage their neuronal EEV platform to identify markers, biomarkers of NGREL 1 and target engagement biomarkers. Our first in human study will be performed in the Netherlands and will be supervised by Leonard Vandenberg. So just before finishing off, I just wanted to highlight 2 people, Mélanie Leboeuf and Stephanie Vargas. Most of the mouse work that I showed you were performed by these 2 really talented graduate students. Mélanie is now a postdoc in Eva's lab in Stockholm, and Stephanie is still with us at BrainEver. Their story is available on bioRxiv, so if you want to learn more about NGREL 1, please go find it and go through it. Thank you so much for your attention, and I'll take any questions.
Thank you very much. It's an intriguing and maybe even paradigm shifting looking at homeoprotein therapy. We have several questions. Gonzalez Adrivakili asks, can you expand on the effects of HEN1 on chromatin?
The effect on heterochromatin. So yeah, so this is one of the aspects that unfortunately we haven't had the time to explore in motor neurons, but we have a number of papers that we've published already where we looked at the effect of ANGREL1 on the survival of midbrain dopaminergic neurons, and what we could show is that this is made possible through the direct effect of human ANGREL1 on the heterochromatin state. So we restore through ANGREL1 treatment major heterochromatin marks like H3K9, H3K27. These are restored pretty rapidly upon treatment at the level of the nigra. So this is something that we're pursuing now in the spinal cord of our animals, and also using patient-derived motor neurons. And we're hoping to be able to report on these progress shortly.
Thank you. Thank you. A question from James Gregory. Can you say more about the cell types of origin of the EN1 that is taken up by motor neurons? You showed us two populations of interneurons that were adjacent to motor neurons, but can you say more about the sources of the HEN1 that gets taken up by the motor neurons?
Well, what I described is our favorite hypothesis right now, because of the, as you said, because of the proximity between the V1 interneurons and the rancho cells with regards to the motor neurons that are nearby. Again, the SCSV approach, which is the overexpression of the SCSV, is local. It's driven by an AAV, so we know that it's acting locally. That the AAV doesn't diffuse that much away from the local astrocytes that overexpress it. So this is really the parsimonious explanation that we really favor at the moment. Should there be any additional source of ANGREL1, we should identify it. We'll look for it and see whether it exists.
Thank you. Ben Brooks asks, is there any consequence of repeated dosing in the non-human primates on sensory neurons? Is that something you've looked at? And in fact, can you comment on the uptake in the sensory neurons?
Yeah, well, so that's something we haven't specifically looked at the uptake in non-human primates in the context of this preliminary tox study. This is something that we're investigating now at the level of the cord. As I said, the pathology analysis that was conducted so far on these limited amount of non-human primates that were injected so far revealed absolutely no negative signal. We had animals that were injected only once. We had animals that were injected 3 times, and we couldn't pick up any signs of toxicity associated with the injection, even in the repeated group injections.
Thank you. Thank you. Two other questions. One is that I think you said that in the mouse model of the hemizygous engrailed mice that there was retrograde axonal degeneration as opposed to degeneration of the whole soma and the axon itself. Can you explain why there would be a pattern of retrograde degeneration?
Yeah, so the reason why we think it might be retrograde degeneration is, but it's twofold. First, we can start to pick up deficits at the level of the neuromuscular junction in the end plates. Yeah. Much before we lose the actual motor neurons and the soma at the level of the ventral horn. And second, this is something that we also observed in a different system, this time looking at the midbrain dopaminergic neurons, where similarly we first saw degeneration in the striatum prior to full impairment at the level of the cell body in the Nigra. So that's the reason why we think it's retrograde degeneration.
Thank you. And the last question is there a timetable on the initiation of the Dutch trial?
Well, we're hoping 2024. We'll hopefully report on that. We'll get a chance to report on that next year.
Thank you.
Next ALS ONE meeting.
Very, very stimulating work. Thank you so much.
Thank you so much.